let's record, share the screen. If you look on the schedule for today, you are supposed to be covering a little bit of chapter 4 and then starting lab 4 on stains. You should read the unknown project instructions file on Canvas for next week. I'll be discussing on Tuesday the unknown project, and we will begin the unknown project on Tuesday. Any questions about that? Where you find the unknown project is on Canvas. Let's see if I can go there. Let's open another one. And I believe it's on week 4 in the summer, but it might be week 5. Come on, show me something. This is really slow today. Week 5 lab. So under week 5 lab, you should read the unknown instructions. This file here, and take a look at it. Any questions about that? Can everybody hear me okay?
I can hear you.
Good.
We're starting that on Tuesday or week 5?
I'm going to be discussing it on Tuesday, and yes, we'll start the unknown project on next Tuesday. Okay? There are a bunch of other files you could read. I'll talk about them on Tuesday, but this one, you need to have that read before I discuss it. All right. So let's return to chapter 4, and I will only discuss it for 20 to 30 minutes. I want to get started on chapter 4. Where do I have chapter 4? I hadn't opened it. I thought I had. All right, like normal, I start out my slides on a chapter, giving you the major goals and a rough outline. You do need to know all of the objectives. I'm only illustrating the major goals. You should know the terms for this chapter. What's a coccus, a bacillus, Barilla? What's a strepto, like streptococcus, staphylococcus, diplococcus? What's a tetrad, a sarcenia, a vibrio, a spirochete, a glycocalyx, a flagella, axiofilaments, fimbriae, pili, peptidoglycan, which you've talked about already, endotoxin, selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid, inclusions, endospores, and then lastly we'll talk about the endosymbiotic theory. You should know on this chapter how gram-positive and gram-negative cell walls differ and how that gives the cells different properties, and then also be able to describe the importance of taxis in bacteria. Any question about what we're covering? Don't hear anything, so I'm moving forward. So when we look at the prokaryotes, you should realize that that's two Greek words meaning free nucleus. Karyotes actually means kernel, but it's translated nucleus. And eukaryotes means true, U, and then nucleus. Prokaryotes have generally one circular chromosome. It's not in a nucleus. When we mean circular, it means where we begin is where we end up on a circle. Eukaryotes have a linear chromosome, so if we look at the X chromosome, it begins in one place and ends in another place. The linear chromosomes in eukaryotes are kept in the nucleus. When we look at the DNA, we'll see that prokaryotes, their DNA is not associated with histones, no histones on, I should say, bacteria. And the DNA of eukaryotes is associated with histones. It's the major protein in the chromosome, the histone protein. Prokaryotes have no organelles, eukaryotes do. I'm talking about membrane-bound organelles. And when I use the term organelle, usually I mean membrane-bound organelle. For a non-membrane-bound organelle, I will use the term cell structure. Like I'll say a ribosome is a cell structure of the cell. I won't say it is a non-membrane-bound organelle. Prokaryotes have peptidoglycan. Well, I should say the bacteria have peptidoglycan in their cell walls, and the eukaryotes have no peptidoglycan in their cell walls. Why I'm making the distinction is that the archaea also don't have peptidoglycan, and you don't really need to know that. And the archaea also have histones, and that's why I need to make the distinction here and there that bacteria do not have histones with their DNA. The prokaryote cells divide by binary fission. The eukaryote cells, they divide by mitosis. And so those are some differences between prokaryotes and eukaryotes. We'll talk about prokaryotes, discussing their cell shape and their growth arrangement. We'll talk about cell structures. We'll talk about movement of material across membranes. We'll then move on to eukaryote, talking about the cell structures, and then lastly, the endosymbiotic theory. When we talk about prokaryotic cell size, you should realize that their average size is fairly small. Most of them range from about 1 micrometer to 3 micrometers, but the larger ones can get up to 10 micrometers in length. They have various shapes and sizes. Prokaryotes have typically 3 basic shapes. The coccus, which is a round cell, a bacillus, or a rod, and there's a good one there, and then the spiral. There are other shapes to prokaryotic cells, but most of them are some variation on one of these 3 shapes. Like we can talk about a curved rod, and some people argue it's half of the spiral. Other people say it's a rod that's bent. The point is, it is one of these 3 shapes. It's just a variation of one of those 3 shapes. So these are the main 3 shapes that all prokaryotic cells have, especially the bacteria. This slide is showing you the different types of spirals. There can be a vibrio, which you could also call that not a spiral but a bent rod. Cholera is actually a vibrio. There's the spirillum, which is the classic spiral. and it's a rigid structure. It has a corkscrew type shape, and it actually moves in a corkscrew type motion. And then there's the spirochete, which is a spiral with many spirals to it. It's more flexible, the spirochete, than the spirillum. There are some prokaryotes that have unusual shape, like Stella, that's a family of bacteria. They're actually star-shaped. And then the Haloarcula archaea bacteria, they're actually, I don't know, square or rectangular shaped. But most cells are not going to be this type of a shape. Most of them are one of those three shapes I gave you earlier. So these are unusual shaped bacteria. Oops, didn't mean to move. I'm trying to just bring it down a little. I can't seem to bring it down. Alright, most bacterial species are monomorphic, meaning they have one single shape. Like all of these are rectangular. All of the spirals are spirals. All of the cocci are round cocci. So most bacteria are monomorphic, single shape. But a few species of bacteria are pleomorphic, where they have many different shapes, even though they're all the same species. Any question about any of that? When we talk about the cellular growth arrangement, the cocci have the most cellular growth arrangements. You can have singlets, or single cocci. You can have two cocci stuck together, and that's called diplococci. And then you can have the cocci stuck together in a row. This we call streptococci. And there's an actual picture of streptococci. You can have cocci that grow as tetrads. Those are groups of 4 cells stuck together. And here you can see 1, 2, 3, 4. It's hard to see here because there's so many of them. And then you can have Sarxinia. That's 8 cocci stuck together. You can also have staphylococci, where you get a clump of cocci. Staphylo means grape, so staphylococci are cocci that grow in a grape-like cluster. You can note that the plane of division determines what cellular growth arrangement the cell can have. If the cell only grows in this plane of division, you'll only get a diplococci or a streptococci. If the cell can grow in this division and that division, then you can get a tetrad. And if they grow in one more division, you can get a Sarxinia. And the staphylococci can grow in all the planes of division. All right, any question about that? Have I talked about everyone? Yep. The bacilli can also be in cellular growth arrangements, but they have fewer arrangements. You can get a singlet or a single bacillus. You can get a diplobacilli. You can get streptobacilli, and this looks sort of like a sausage link. You can also get a, streptobacilli, where the cells are not attached on the long end, they're attached on the short end. And this we call a palisade. If you rotate that 90 degrees, meaning that this is the bottom and this is the top, it looks like a palisade fence. And if you don't know what a palisade fence is, it was discussed in Mark Twain. And I don't remember if it was Tom Sawyer or Mark Twain. Probably Tom Sawyer had to paint the palisade fence. It's just a board stuck together to make the fence. And they used to be very common in the past. I don't know what era Mark Twain was, but in the past it was a very common fence. You can still see them today, palisade fences. All right. I should note that Coccobacillus is not a cellular growth arrangement, and I don't know why it's here, although this is a Diplobacilli. Coccobacillus is a shape, and what you have is, if you have a bacilli that's rounded on the edges, and you make it shorter and shorter, it will eventually become a Coccobacillus. A Coccobacillus is a cell in between a bacillus and a cocci. Any question about that? So this is a variation of a shape of the cell, not a growth arrangement. And I don't know why that's in this slide. Another thing I need to mention is that when we talk about the term bacillus, take a look and see if it is capitalized or if it is lowercase. If it is capitalized, we mean the family of Bacillus, like Bacillus megaterium. So it's a family of bacteria. If it is a lowercase b for Bacillus, we mean the shape of the bacteria is in a rod. Okay? And don't get confused, where is it? With the Streptococci, which is a cellular growth arrangement. There is also a Streptococcus, and that is a family of bacteria. The Streptococcus will be a capital S, and although the Streptococci here is with a capital, usually when you see it in a sentence and it's referring to the growth arrangement, it'll be a lowercase s. Any question about any of that? All right, so don't confuse the family name with the cellular growth arrangement or the shape of the cell. Here we're looking at some typical structures to a prokaryotic cell, and everything in red is a structure that you'll find in all prokaryotic cells. The cytoplasm, you can actually find that in all cells. Ribosomes, also you can find that in all cells. The plasma membrane, you can find that in all cells. And then the DNA, you can find in all cells. Now, the DNA of a prokaryotic cell is simply floating around in the cytoplasm. And there is no distinction between where the DNA is and where the cytoplasm is, because there's no membrane separating them like there is in a eukaryotic cell. So it's wherever the DNA floats, that is where we call the nucleoid region. It's where the DNA is in a prokaryotic cell. And when you look at it, the nucleoid region does stain differently than the cytoplasm. But, for example, in this region of the cytoplasm, there's no DNA, but the DNA can move over here, just float over there. And then that would be a nucleoid region. Any question about that? So I guess the point is the nucleoid region can move around. It's wherever the DNA floats in the cytoplasm. And usually the DNA is in solution of the cytoplasm. Any question about that? All right. If not, let's move on to the next slide. Well, I can mention we'll talk about the cell wall, about capsules, inclusions, flagella, and the plasma membrane. I guess we'll briefly talk about Fimbriae too. The glycocalyx, also pronounced glycocalyx, is something that the cell makes and secretes, but we call it outside the cell because it's outside the cell wall. It's usually sticky, and there are 3 types of glycocalyxes. The capsule is a neatly organized layer outside the cell wall, and you can see the capsule under the light microscope. The main portion of the importance of a cell having capsule is the capsule can prevent the bacteria from being phagocytized by a phagocyte. So the capsule can prevent or help prevent phagocytosis. Capsules can be seen by a light microscope, but a slime layer cannot be seen by a light microscope. A slime layer is mostly water, and it's something secreted by the cell, meaning it's a glycocalyx, and it's very unorganized, a loose structure, and you can't see it with a light microscope because it's mostly water. Okay? Any question about that? To see the slime layer, you'd have to look at the cell with an electron microscope. There is a 3rd type of glycocalyx, and it is only seen in cells that make a biofilm. The extracellular polysaccharide of the biofilm is made by the bacteria and secreted outside the cell, and that is considered a glycocalyx. The difference is this glycocalyx will make a biofilm. It'll have lots of bacteria in it, and it would be impossible to determine which region of the extracellular polysaccharide was made by this cell and what region was made by that cell. Okay? We probably won't discuss biofilms very much in this class. A flagella is a structure on a prokaryotic cell that is mostly outside of the cell, mostly outside of the cell wall, but there is a small portion of the flagella which is connected to the cell wall and connected to the plasma membrane. And those are called the basal body. The flagella is a whip-like structure that can move, and if the bacteria can swim or move in a liquid media, that means the bacteria has a flagella. And why can I say that? Because the flagella is the only structure that prokaryotic cells have to allow them to swim or move in a liquid media. So the flagella is used for locomotion, and it's made of the flagellin protein to make the flagella, which this portion is called the filament. Any question about the flagella? Now, don't tell me that flagella is only seen in prokaryotic cells, because some eukaryotic cells do have flagella. In humans, the cells of the trachea have flagella, Or is that cilia? Just a minute, let me think about that. Sorry, the cells of the trachea have cilia, which is something different. I'll talk about it later. The only cell that humans have that have a flagella is the sperm cell. Is that right? I think that's right. You can have 4 basic arrangements of flagella in the cells. And I'm not going to test you on this, but you need to know for one of the labs. So one of the questions in the lab. Monotrichous means the flagella is on one end. Amphitrichous, the flagella are on both ends, and there's only one. Lophitrichous, multiple flagella on both ends. And then peritrichous, the flagella are all over the cell, and there's many of them. I said, don't remember the names, but you'll need to know this for a question on a lab. Now remember, if the bacteria cells move, they must have a flagella, and cells can rotate or run or tumble using the flagella. Taxis is the movement toward or away from a stimuli. And for bacteria, we can have 2 different types of taxis. There can be chemotaxis, where the bacteria either move toward or away from a chemical stimuli. And then there's phototaxis, where the bacteria can move either towards or away from the light source. Now this bacteria is moving away from the light source, but it is moving by phototaxis. Any question on any of that? All right, if there's no further questions, I think I will stop here with the lesson and get started on the lab. Lab 4 we're doing. I already got it open. Never mind. All right, Lab 4. Staining techniques. There are differential stains, and special stains. You need to read the textbook on these pages here about the different stains. And you should read this before you begin the lab. In today's lab, there will be video clips and I think a virtual lab. The learning objectives. You should know how to prepare a bacterial smear for staining and realize that it's always done before you're staining the slide. And it's usually considered something separate, and we call it a bacterial smear. You should also understand the principles and the procedures used for performing a simple stain, a differential stain, and a special stain. Be able to interpret the results for a simple stain, a differential stain, and special stains. And then lastly, be able to define the following terms: simple stain, differential stain, special stain, what's a coccus, bacillus, spirillum, peptidoglycan, what is a gram-positive cell, a gram-negative cell, what's an acid-fast stain, what's a primary stain, what's a mordant, what's a decolorizer, what's a counterstain, what's a capsule, what's an endospore, and what is a flagella. All right, any question about any of that? Oops, too far. Because of their extremely small size, Bacteria are essentially transparent. Therefore, in order to see them under the light microscope, it is necessary to stain them before they can be observed. So when you're going to stain a slide, you first have to make or prepare a bacterial smear. How you do that is you add a drop of sterile water on a clean slide, and then you add the bacteria to the drop of water. You do not put on a loop full of cells where the entire loop is full of cells. What you do is only put on a very small portion of that loop, maybe 1/8 of the loop. And this is obviously a pen, but I'm trying to pretend it's a loop. Okay? I don't have any loops with me here. You then smear the bacteria all over the surface of the slide. Ideally, you want the bacteria to be 1 layer thick. You then allow the slide to air dry, and then you heat fix it. The air drying and heat fixing step allow the bacteria to stick to the glass. Otherwise, without those 2 steps, the cells, the bacteria, would wash off the slide in a washing step, and most staining procedures have a washing step. A simple stain is where you stain the cells using only one stain. And actually, it's not a basic stain. It usually is a basic stain, but it can be a basic or a negative dye. It can be either. As long as you're only using one, it's a simple stain. A differential stain usually uses 2 or more dyes and then provides you additional information about the cell. So in the simple stain, all cells will be stained the same color. In a differential stain, you'll see differences between different cells, and they can be stained a different color. And usually they are stained a different color. And then lastly, there's special stains. Their purpose is to stain special structures in the cells. We discussed this in the lab, some different morphologies of bacteria. You can get singlet cocci, diplococci, tetrad cocci, Sarxenia, which are 8 cocci, streptococci, where the cocci grow in a row, and then staphylococci, where the bacteria grow in a clump. With the bacilli, you do not get that many arrangements. There are singlet bacilli, there are diplobacilli, there are streptobacilli, and then there's palisades, which are similar to a streptobacilli. I never mentioned it before. What's the cellular growth arrangements you can see with the sporulum or a spirochete? Does anybody want to guess? Come on, somebody guess. Where she can be is wrong. Well, why am I not discussing it with the spirals? Nobody wants
Honestly,
to
no
guess.
idea.
What?
Honestly, no idea.
Okay, it's because they only have one cellular growth arrangement, and that is singlets. That's why we didn't discuss it with the sporulum. Okay? All right, so let's talk about the differential stains. The most commonly used differential stain was the Gram stain, which was invented in 1884. You don't need to know the year, but you should know that Dr. Gram, Hans Christian Gram, invented the stain, and that's why you should make it a capital G whenever you spell out Gram stain. It is a differential stain in that it allows you to see differences in the cell walls of bacteria. Crystal violet is used as the primary dye, and the bacteria that retain the crystal violet are classified as gram-positive cells, and they will be purple. And then there are other cells that do not retain the crystal violet, and these cells we call gram-negatives, and they would be the pink staining cells here. Okay? Any question about any of that? The gram reactions are based on the ability of the cell wall to retain the crystal violet. Gram-positive microorganisms contain a thick peptidoglycan layer, whereas gram-negative bacteria have a thin peptidoglycan layer. So there we see a gram-positive cell with a thick peptidoglycan layer, and here we have a gram-negative cell, and it has a much thinner peptidoglycan layer. The gram-negative cell, we should mention, has a lipid bilayer on the outside, which is called the outer membrane of the cell wall. Come on. I hit the wrong button. Some gram-positive bacteria lose their ability to retain crystal violet and are easily decolorized. Sometimes they're called gram-variable cells because when you stain them with the gram stain, some of the cells will look pink and some of the cells will look purple. Like in this stain here, which is a gram stain of one species of bacteria, so they should be staining the same, and as you notice, they're staining two different ways. Some are staining like gram-negative, some are staining as gram-positive. This is especially seen with the Mycobacterium because they have a lot of waxy substances in their cell wall, and it's called mycolic acid. And that mycolic acid does not readily take up any water-soluble molecule, including the dyes of the gram stain. Okay? And that's probably what we're looking at here. But I notice that when we look at these cells, that I can detect a difference between this gram-negative cell and this gram-negative cell and these gram-positive cells. What can you determine, like from this gram-negative cell or that gram-negative cell, in comparison with this gram-positive cell and that gram-positive cell? What looks different between those cells?
The color?
Well, yeah, the color, obviously. I'm talking about what looks different besides the color. Come on, somebody's got to be observant enough to notice that. What does this cell look like right here? It's particularly true of that one, but also that one, and to a lesser degree that one. I got the pointer on it. Okay? Nobody noticed it? This cell sort of looks like a ghost. It's not totally clear. The boundaries of this cell are very clear, very sharp. The cell is very darkly stained, and so it shows up very well. This cell and that cell looks a little bit like a ghost. In fact, this one right there in the middle, it looks like you can't even see part of it. The boundary is not sharply demarcated. And in fact, the boundaries may be falling apart. And that is what I think's happening here, and that is the gram-negative cells are older cells, and these cells are dying. That one is probably dead, okay? That one is either dead or it's shortly going to be dead. And this cell is a freshly growing cell. So is that one. And that is all of the gram variable slides I've ever looked at. And I asked the student, how old are these cells? Because some of them are staining gram positive, others are staining gram negative. And the student tells me, in every case, the student has told me, oh, those cells are old. They're 2 weeks or older, okay? The gram stain should not be used on cells that are that old. If you use the gram stain on old cells, the gram stain will not stain correctly. The gram stain should be used on only cells that have been grown for 2 days or less. And if you look at the Mycobacterium for cells that are grown for 2 days or less, every time I've looked at it, I've only seen gram-positive Mycobacteria. Okay? It's only on the old Mycobacteria that we see the gram variable. And you shouldn't be using the gram stain on old cells. Okay? Because it doesn't work correctly on old cells. And the reason why is, if this cell has a hole in it, the crystal violet can leak out the hole. And then it's not going to be staining purple if it's a gram positive. All right? Any question about any of that? So when you're doing the gram stain, you should always do it on freshly grown cells. And that's cells that haven't grown for more than 2 days. When you're making a smear preparation for especially the gram stain, but always, avoid using old cells. Make the smear thin. If it's not thin, you're going to have trouble getting dye into the lower cells. And then if the dye is supposed to wash off from the lower cells, it's going to have trouble washing out of the lower cells. So make the thin smear. Heat fix the bacteria only after the smear is air dried. Otherwise you will cause disruption of the cells. The water will boil in the cell and pop the cell. And then avoid overheating the cells, because you can change the shape of the cells if you overheat the cells. Do not over decolorize or under decolorize the cell. All right? Any question about any of that? Okay, those are some tips for doing the gram stain. The acid-fast stain is a stain we use to stain bacteria like the Mycobacterium in the Nocardia, which have waxy substances in their cell walls. This molecule is mycolic acid, and it's waxy, and it does not easily retain water-soluble stains. But these cells are resistant to the acid-fast stains, and so they'll stain with an acid-fast stain and retain that stain, whereas other bacteria will lose that stain. And these bacteria we call acid-fast bacteria. There are 2 different stains we'll look at, the Zeal-Nelson stain and the modified Kenyon stain, and they're both acid-fast stains. What they do is, an acid-fast cell will retain the acid-fast dye and will be pink in color. And they call that acid-fast positive cells. And then the other cells do not retain the acid-fast stain, and they will be bluish or greenish. All right, any question about that?
Is calling it a acid fast bacteria negative the same as calling it non-acid fast bacteria?
If it's not acid-fast bacteria, it would be the same thing as non-acid-fast bacteria. Is that what we're saying?
Yeah, is negative the
Oh,
same?
yeah, negative is not acid-fast.
Okay, thank you.
Yeah. Positive is acid-fast. All right. So those are the two main differential stains, the Gram stain and then the acid-fast stain, which show differences between cells. There are also special stains, and special stains are stains that we use to specially stain a structure of the bacteria. Like we can use a negative stain to stain the glycocalyx. And usually what we're looking for is the capsule, because the slime layer cannot be seen under the light microscope, even with the stain. You can't see the slime layer, but we can see a capsule when we use a special stain. You could also use a special stain to stain a flagella. That's a structure that is not normally stained by the staining process, and so you will not normally see it under the microscope unless you use a special stain called the flagella stain to stain the flagella. And then there's the endospores or some other special structure. You can use a special stain to see the special structure of the cell. Okay, capsules are important because they can add to an organism's virulence. So generally speaking, organisms that infect us, meaning bacteria that can infect us, they are more virulent if they produce a capsule. And these are some of the species that can produce a capsule. And I think you know Klebsiella pneumoniae, Haemophilus influenzae, Bacillus anthracis are all serious diseases, and they produce a capsule. Any question about any of that? I could point out that the capsule makes the colony of bacteria when it's grown on a petri dish look mucoid. When we look at the capsules, oops, I lost that. When we look at the capsules under the microscope, they will be non-staining. And then the background will be lightly staining, and the cells will be staining, depending on the capsule stain. This one stains the background lightly, and the cells darkly, and the capsules not at all. If you're using India ink, it stains the cells lightly, and the background darkly, and the capsules not at all. And this is capsules of Klebsiella pneumoniae. And that's not India ink. It says it's India ink, but this is not India ink. All right. There's also endospore staining. And endospores are structures that cells make to survive a harsh environment. Endospores are metabolically inactive, hardy structures produced by some bacteria as a defense to allow the cells to survive a harsh environment. And generally, there's only two families of bacteria that produce endospores, the family Bacillus and the family Clostridium. When the environment becomes unfavorable, the bacteria will start making an endospore. And the cell that makes the endospore we call the parent cell. And the parent cell will eventually die from the unfavorable environment. But if it makes the endospore before it dies, that endospore will then survive the harsh environment. And the endospore is just waiting for the environment to become favorable for growth. And then the endospore will germinate and the parent bacteria will come back, meaning we call that a vegetative cell, a cell which is actively growing, is actively metabolically active, and is reproductive. This table is giving you some of the cells which form endospores. And you'll need to use this table to answer at least one of the questions, maybe some of the questions, in the lab worksheet. All right, endospores stain differently from the parent cell or a vegetative cell. So when we use the Schaefer-Fulton endospore stain, it stains the endospores green, and if you notice, this cell right here has a red on this side of the cell and a red on that side of the cell, and the endospore is on the inside of the cell. The same with that one. What's happening is the red cell is making the endospore, and the endospore is being formed inside the parent cell. That's why it's called an endospore. Any question about that? Anyways, endospores stain greenish, and the vegetative cell or the parent cell stains red. All right, the flagella stain is a stain used to show flagella. Flagella are long thread-like or whip-like appendages that are used by the cell for movement. However, they're too small to be picked up by most stains, and so when we look at the cells under the microscope, we will not, under the light microscope, we will not see the flagella unless you use the flagella stain. Usually the flagella stain coats the flagella many times, and then it makes it visible under the light microscope. You'll need to know this table here, or this whatever figure here, to answer one of the questions, but this is not something I'm going to test you on, and that is the name of the arrangement of the flagella. Okay? And when you look at the flagella under the microscope, you can note here, let me blow that up, that some cells have different flagella arrangement. Like this one here is clearly peritrichous, but this one here, is that one or two? That one's clearly two flagella, so that would be I don't have that one here. A variation of amphitrichus. Okay? So what do you do when you have multiple cells staining differently with the flagella? Well, these are all the same species, and what we do is we go on the highest flagella arrangement. So the arrangement of flagella here would be peritrichous, because this one is peritrichous, that one is peritrichous, that one is peritrichous, that one's slightly peritrichous, and this one is not peritrichous, but it is putting out the flagella. With more time, more flagella will be made, and this one will eventually become peritrichous. Any question about any of that? All right. So know the terms of this chapter. You don't need to pay attention to the references, but do laboratory exercise 4. Remember to write your answers in the worksheet. In the future, if anyone turns on the lab manual, I'll give you a point deduction for the lab. So only turn in the worksheet. While you're not physically going to be performing any of these activities, you must understand how to prepare bacterial smears for staining. You must learn how to prepare a bacterial smear for staining, and we have 2 videos on that, preparing the bacterial smear and then the heat fixing. Watch those videos and read through this. And then go through the procedure on simple staining. We have a video of that. And you do need to answer some questions here in the worksheet. Let me show you the worksheet. Oh, too far. So you do need to answer question procedure 2, question 3A1 and 3A2. You need to answer those 2 questions, and then 3B. Okay? So answer them in the worksheet and turn in the worksheet. Where was I? Ooh. Then do procedure 3 on the gram stain. We have a video clip on a gram stain, and we have a virtual lab on the gram stain. Let me open that, make sure it works. Come on. Ah, wrong button. The virtual lab is where you will be able to do the gram stain on your computer. Oh, let me run it from the internet. It's not lab 6, it's lab 4. It's hidden. Let's open that in a new tab. There you go. So it will take a while to load. Click continue.
Hi, Lab Assistant.
Jeez.
Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria. All yogurt has bacteria in it, since yogurt is made by...
All right, so that works. That was a little loud for my earphones. Hopefully that wasn't too loud for you. Let me turn the volume down. I remember I turned that way up because I was having trouble hearing a student. All right. So we have a virtual lab on the gram stain. Watch that and work with it. It'll allow you to do the gram stain on your computer. And then read through these different steps, go through the procedure, and then answer these questions in the table. Answer the questions here, not in the lab module, do it on the worksheet. And then do procedure 4, acid-fast bacteria. There's a little video to watch. Read through the procedure. And then there are questions here. You need to mark, interpret whether the organism denoted by the arrows are acid-fast positive cells or acid-fast negative cells. And you can either write which ones are positive, which ones are negative, or you can put it right here what it is, okay? Either way. And then do procedure 5. It's looking at special stains. We do have a video clip on the endospore staining and a negative staining. And then we have 3 slides, A, B, and C here, which are showing you the special stains. Answer the questions for slide A, answer the questions for slide B, and answer the questions for slide C. Any question about any of that? You do have to understand the slides and the stains to answer these questions. And then lastly, we have 2 case studies for you. To answer these, give me the most probable diagnosis. You must name the species which is causing the infection. So in case study 1, there's some information about the patient, some tests that were done on the patient, an x-ray, a sputum smear, and the Zeal-Nelson stain. Tell me what the bacteria is which is causing the patient's illness. And then in patient case study 2, some information about the patient, some physical information about the patient, the Gram stain of the patient, and the Schaefer-Fulton stain. And then give me the species which is causing the infection. Okay? If you don't give me the species, it will be wrong. So you have to give me the species name. Okay? All right, any question about any of that? If there's no questions, go ahead and start working on the lab. I will be here until 8:20 to answer questions, or until the last student logs off.